Hello guys, today we're going to be talking about the circulatory system, specifically about blood. This is chapter 18. As I just said, the circulatory system consists of the heart, the blood vessels, and blood. And for the cardiovascular system instead, we have only the heart and the blood vessels. Remember, among the functions of the circulatory system, we have the transport of gases such as oxygen and carbon dioxide. We're transporting nutrients, waste, chemicals such as hormones, and stem cells. The circulatory system, another function is going to be protection. Remember, inflammation, limiting the spreading of the infection, destroying organisms such as bacteria and viruses and cancer cells, neutralize toxins, and initiate clotting or coagulation is going to be another function of the circulatory system. And we have, as another function of the circulatory system, the regulation. And it's going to be the homeostasis. If you remember that concept, it's going to be the internal balance. Then we're able to regulate the fluid balance inside our blood vessels, stabilize the pH of the extracellular fluid, and we are able to control the temperature as well. Well, what kind of tissue is the blood? If you remember well, IP1, blood is the connective tissue, and it has a fluid part, which is the ground substance or the matrix of the blood, which is a fluid part, a liquid part, and is the most abundant part, which is 55% of the whole blood. The other part is going to be the form elements or cellular elements. Then we have 45% of the solid part of the blood. Then 55% is going to be liquid or fluid, and 45% is going to be solid. And among these solid, we have three kind of form elements. We have erythrocytes or red blood cells. We have leukocytes or white blood cells, and we have pieces of cells, pieces of cytoplasmic parts called platelets. Then erythrocytes are for transporting gases such as oxygen and carbon dioxide. And we have inside of these cells, by the way, it's the only mature cell without a nucleus. It's an enucleated cell in our body. And the leukocyte cells is for protection. When we detail some leukocytes, we're going to check the protection function because it's the immune system protection. And another function we have through platelets, and we have the coagulation or clotting of the blood. It's because of the platelets. Then after, when we have whole blood and we put a tube here for the centrifugation, we're going to separate this liquid part and this solid part. The solid part is going to be the form element or cellular elements, and the plasma is going to be the fluid part. And after, we're stimulating the coagulation or clotting of these proteins, because we have in this part some proteins for coagulation. We're going to get some clotting proteins such as fibrinogen, and we're going to separate the clotting protein from the serum. Then serum is going to be the plasma without the clotting proteins. Going in detail about the form elements or cells in our blood, we have some, the most abundant cells are the red blood cells. These are erythrocytes. They have this shape, and they are again enucleated. Then we have pieces of cytoplasmic parts of the thrombocytes called platelets. and these are in charge of coagulation. Then we have these bigger cells. All of them are white blood cells or leukocytes. And we have, regarding this part, we have some kind of white blood vessels. And we can divide regarding if we have granules like this one, and this one, and this one, or if we don't have granules like this one and this one. If it's one cell with granules like this one, is called granulocytes because of the granules. If it's a cell without granules, it's called agranulocyte. Among the cells with granules, we have the most abundant cell with granules is going to be the neutrophil. The neutrophil is going to be this one, and is the main character for our defense against infections and toxins. This is called neutrophils, and because they don't have a definite color for their granules. Then we have another color for granules, another kind of granulocyte called eosinophil, and it's because we have red granules after their coloration. We have red granules. And the other kind of granules is going to be blue granules. That's why we have the other kind of granulocytes going to be the basophils. Then we have neutrophils, eosinophils, and basophil. And for not granulated white blood cells, not granulated leukocytes, we have these big one with a nucleus with the shape of a half of a moon, is called monocytes. And we have these small leukocyte, and almost all of it occupied by the nucleus is going to be the lymphocyte. Then we have granulocytes and agranulocytes. When we separate our blood in the liquid part and the solid part after centrifugation, we're going to get this liquid part divided from the solid part. And if you see here, you can see 99% of the total volume of this one is going to be red blood cells or erythrocytes, and they are the most important cell of the blood. And we have a buffy coat here, right here, which is about 1%, maybe 2% of leukocytes and platelets. And plasma is going to be again 55% of the whole blood. Here in plasma, we're going to have 92% of water, and we're going to have some proteins, three kind of proteins. Actually, we have albumin, gamma globulins, and we have fibrinogen. And we have another things here, such as proteins, nutrients, electrolytes, waste from nitrogenous, such as urea, some hormones and gases diluted here. Remember, to separate our blood, we centrifugate our blood and we separate the solid part from the liquid part, and the liquid part is plasma. And after stimulating the coagulation or clotting of the blood, we can separate our serum from the clotting patterns. That is, serum is going to be always plasma without the clotting patterns, proteins. Then we have serum when we remove all of these proteins in charge of the clotting of the blood from the plasma. Again, remember, from plasma we have three kind of proteins. We have albumins, globulins, and fibrinogen in charge of clotting. Besides proteins, we have some nitrogenous compounds, some free amino acids from the diet, if we're eating a lot of proteins, and some wastes, and such as urea, which is the most abundant waste from the protein metabolism. We have some nutrients as well, could be in plasma. We can have glucose, vitamins, fats, or lipids, cholesterol, phospholipids, and minerals. And we can have some, the main cell for transporting gases is going to be the red blood cell, but we can have some dissolved oxygen and carbon dioxide and nitrogen inside the plasma. And another important component of plasma is going to be electrolytes, but sodium is going to be making up to 90% of the plasma cations here. Remember that. Regarding blood, we have some normal values, some properties we need to know. For example, regarding the volume in an adult, it's going to be about 5 liters. Regarding the temperature, it's going to be about 38 degrees Celsius, about 100 degrees Fahrenheit. Our normal value of pH for blood is going to be 735 to 745. If you remember AP1, below 735 is going to be acidosis, and above 745 is going to be alkalosis. Remember the viscosity, we're going to have more viscose than water, and the whole blood is going to be 4 to 5 times more viscose than water. And plasma, which is the blood without cells, without the 4 elements, is 2 times more viscose than water. Regarding another important value is going to be the osmolarity. And this is about the oncotic pressure. And we have around 280 to 296 milliosmoles per liter. The mean salinity is going to be, about mainly because we have salt in our blood, and mainly from sodium. Remember, 90% of the electrolytes, it's 90% of the cations is going to be sodium, and we have 0.9% of salinity. That is why we receive, when we need some hydration, some IVs, then the most frequent solution that we use is normal saline, or 0.9%. The hematocrit is going to be the packed cells volume, and it's going to be a little bit higher in males than females. It's going to be about 3 times the hemoglobin, and it's going to be the packed cell volume. Remember, the red blood cells are the most abundant cell in our blood. And regarding the hemoglobin, we have 12 to 16 grams per deciliter, and it's going to be a little bit higher for males, 13 to 18 grams per deciliter. Below these values, we consider anemia. Anemia is going to be the reduction of hemoglobin in our body. Platelets count, it could be around 130,000 to 360,000 per milliliter, and we're going to have maybe a different counts regarding the textbook. And regarding the white blood cells, is the less common cells of blood. It's going to be 5,000 to 10,000 cells per milliliter. Regarding these values, when we have below 5,000 on the white blood cells count, we're going to have leukopenia. When we have more than 10,000, we're going to have leukocytosis. Again, below 5,000 is going to be leukopenia, and above 10,000 is going to be leukocytosis. Regarding the platelet count, if we have below 130,000, it's going to be thrombocytopenia. If we have above 360,000, we're going to have thrombocytosis. Then we have thrombocytopenia and thrombocytosis. Below these values, again, we have anemia. We have anemia below these values. And when we have a lot of red blood cells When we have the red blood cells very high, then we have polycythemia. Regarding proteins, albumin is going to be responsible for the colloid osmotic pressure. The plasma is going to be like a magnet for the blood. We have our blood inside the blood vessels because these proteins are working as a magnet. Then they're keeping our blood inside our blood vessels. And we have some globulins, 36% of the proteins are globulins. Then we have alpha globulins, beta globulins, and gamma globulins. Remember, these are the antibodies, very important, and we're going to be talking about that later. Fibrinogen for clotting is going to be around 4%. Here you can see some other components of plasma. Remember, 92% is going to be water, and some other part is going to be proteins. Albumin is going to be 60% of it. 36% is going to be globulins. We're going to have alpha globulins, beta globulins, and gamma globulins. These last are the antibodies. And fibrinogen is going to be about 4%, and this is going to be in charge of clotting. Then nutrients, electrolytes, nitrogenous waste. The most important one is going to be the urea, again, and some other components such as diluted gases. The first cell we're going to regard now is going to be the red blood cells. Remember, they are the most abundant cells. This is the shape, and you can see these are the unique cell, the only cells without a nucleus. They are enucleated. It's the most abundant cells for millions and millions, and you can see here a lateral view inside the capillary. Most of it is going to be occupied, the red blood cell is going to be occupied by hemoglobin. And here you have the hemoglobin structure. You have 2 beta chains, 2 alpha chains, and 4 groups M's. This is the molecule of hemoglobin. Hemoglobin is its molecule. Very important. 2 beta chains, 2 alpha chains, and 4 groups M's. About the erythrocyte or the red blood cell. Regarding the origin of the red blood cell, we have here the hematopoiesis, which is the origin of the formation of the red blood cell. Then the origin is going to be embryologically, considering this cell, which is the stem cell or hematopoietic stem cell, is going to be the origin for the red blood cell. Then we're going to have some precursor cells called erythroblasts, and these erythroblasts, they have a nucleus. And then finally, these are the directly precursor cells before the whole maturation is going to be reticulocytes. These are the last cells before red blood cells mature with nucleus, actually with pieces of nucleus. And then at the end, we're going to have a mature cell able to transport gases, and it's going to be with no nucleus. These are erythrocytes. Then this is going to be the origin of the erythrocytes. Remember, the concepts in hematopoiesis is going to be the formation of the red blood cell. All of these are immature red blood cells with nuclei, and these are mature red blood cells without nuclei.